Hello everyone, my name is Roseanne Balsa. Today I will be presenting you the chapter 15 topic, which is carbohydrates of physiological significance. And let's go ahead and start with the introduction. So the topics that are going to be covered in this PowerPoint and presentation will be carbohydrates. I'm going to talk to you about the definition, classification, examples, and importance of carbohydrates. Also, the glycobiology definition. We will talk about the most important carbohydrate, which is glucose. I will give you the structure and the isomers. I will present to you a table of pentoses and hexoses of physiological importance. Um, we will learn about glycosides, amino sugars, polysaccharides, glycoproteins, the definition and examples. And lastly, we're going to talk about the presence of carbohydrates in cell membranes and lipoproteins. We're going to start learning about the definition of carbohydrates, which is that carbohydrates are essential micronutrients that are the primary source of energy for humans. They are ingested in the form of simple carbohydrates like monosaccharides and disaccharides, or complex carbohydrates like oligosaccharides and polysaccharides. So in simple terms, carbohydrate is the preferred energy source of the brain, red blood cells, and muscle cells during intense activity. Next, we're going to learn that glucose is the most important carbohydrate, and in fact, it is absorbed into the bloodstream as glucose formed by the hydrolysis, and other sugars are converted to glucose in the liver. Now we will see that glucose is the major metabolic food for mammals and the universal food for the fetus. So we have to say that glucose is the only food used by the fetus, highlighting its physiological importance. Now we're going to talk about the definition of glycobiology, which is that glycobiology is the study of the role of sugars in health and disease. The glycome is the entire complement of sugars of an organism, whether free or in more complex molecules. Glycomics is the comprehensive study of glycomes, including genetic, physiological, and other aspects. So here we need to say that understanding glycomics is the most important. It's important because it helps us understand the role of complex sugars like glycans in biological processes like cell signaling, immunity, and development. Also, we need to say that abnormal glycan structures are linked to diseases such as cancer, infections, and genetic disorders, making glycomics crucial when it comes to diagnosing diseases in patients. So in this slide, we're going to learn about the classification of carbohydrates. We're going to talk about some of the definitions of them. So to start, we're going to start with monosaccharides. And we have to say that those are sugars that cannot be hydrolyzed into simpler carbohydrates. They might be classified as trioses, tetroses, pentoses, hexoses, or heptoses, depending on the number of carbon atoms. Also, they can be classified as aldoses or ketoses, depending on whether they have an aldehyde or ketone group. Next, we're gonna read the definition of disaccharides. And those are condensation products of two monosaccharide units, for example, lactose, maltose, isomaltose, sucrose, and trehalose. Oligosaccharides are condensation products of 3 to 10 monosaccharides. Most of them are not digested by human enzymes. And lastly, we're going to talk about polysaccharides. and they are condensation products of more than 10 monosaccharide units. And some example of them are the starches and dextrins, which may be linear or branch polymers. Also, polysaccharides are classified as hexosanes or pentosanes, depending on the constituent monosaccharides. In this slide, we're going to see the structure of glucose, which can be represented in three ways. And here I have an image of the three different ways that it can be represented. So in letter 8, we're going to see the straight chain structural formula, aldehydrols, which can account for some of the property of glucose, but a cyclic structure is thermodynamically favored and accounts for other properties. Next, we're going to see the cyclic structure, which is normally drawn as shown in figure B, the Hayward projection, in which the molecule is viewed from the side and above the plane of the ring. The bond nearest the viewer are bold and thickened, and the hydroxy groups are above or below the plane of the ring. And lastly, we're gonna see in letter C, we're gonna see the ring in the form of the share. And here we have the image. So this slide, we're gonna talk about the more important types of isomers that are found with glucose. And the first type is the D and L isomerisms. The designation of a sugar isomer as the D form or its mirror image as the L form is determined by its spatial relationship to glyceraldehyde. The number 2 is the presence of asymmetric carbon atoms that also confers optical activity on the compound. And when a beam of a plane polarized light is passed through the solution of an optical isomer, it rotates either to the right or to the left. The number 3 is the pyranose and furanose ring structures. In this case, the ring structures of the monosaccharides are similar to the ring structures of either pyran or furan. We're going to continue talking about the type of isomerisms. And in this slide, we have number 4, 5, and 6. And in number 4, it refers to the alpha and the beta anomers. In this case, the ring structure of aldose is a hemiacetal, since it is formed by the reaction between the aldehyde and an alcohol group. Number 5, epimers. In this case, isomers differing as a result of variations in configuration of the OH and the H on carbon atoms. 2, 3, and 4 of glucose are known as epimers. And lastly, we have number 6, which is the aldose and ketose isomerisms. In this case, fructose has the same molecular formula as glucose, but differs in that there is a potential keto group in position 2, whereas in glucose, the CRE is the potential aldehyde group in position 1. Okay, so in this slide, we have the pentoses of physiological importance. This is a table that includes the different type of sugars, the sources, and the biochemical and clinical importance. So the first one that we have is D. rabies. The source in this case will be nucleic acids and metabolic intermediate. And the biochemical importance will be the structural component of nucleic acids and coenzymes, including ATP and AAD and flavin coenzymes. The next type that we have is D. raveolus. The source is metabolic intermediate, and the importance is the intermediate of the pentose phosphate pathways. Next, we have the D-arabinose. The source is plant gums, and the importance is the constituent of glycoproteins. Next, we have D-xylose. The source in this case will be plant gums, proteoglycans, and glycosaminoglycan. The biochemical importance would be the constituent of glycoproteins. And lastly, we have L-cellulose. The source will be the metabolic intermediate. And lastly, the biochemical and clinical importance will be the excreted in the urine in essential pentasodium. So in this slide, we have another table that talks about the hexoses of physiological importance. And here we have the different type of sugars. We have the sources, the biochemical importance, and the clinical significance. So the first type of sugar that we're going to be talking about is the D-glucose. In this case, the sources are the fruit juices, hydrolysis of a starch, cane and beet sugars, maltose and lactose. And the biochemical importance is that the main metabolic fuel for the tissue's blood sugar. So the clinical significance is that this type of sugar is excreted in the urine, and in poorly controlled diabetes mellitus as a result of hyperglycemia. The next type of sugar is the D-fructose. The sources are fruit juices, honey, hydrolysis of cane or beet sugar, and inulin. Also, the enzymic isomerization of glucose syrups for food manufacture. The biochemical importance in this case is that it really metabolizes either via glucose or directly. And the significance is that hereditary fructose intolerance leads to fructose accumulation and hypoglycemia. Next, we're going to talk about the D-galactose type of sugar. And the source in this case is the hydrolysis of lactose. The importance is that it readily metabolizes to glucose, synthesized in the mammary gland for synthesis of the lactose in milk. The clinical significance is the hereditary galactosemia as a result of failure to metabolize the galactose, which leads to cataracts. The last type of sugar that we have is D-mannose. The source is the hydrolysis of plant mannan gums, and the biochemical importance is the constituent of glycoproteins. And here we see that there's nothing for the clinical significance. So this is all for this for the hexoses. Now we're going to be talking about glycosides, and here I have some important information about it. The first point that I would like to discuss is that glycosides are formed by condensation between the hydroxyl group of the anoramic carbon of a monosaccharide and a second compound that might be another monosaccharide, or in the case of an I-glycone, not a sugar. Also, we need to know that glycosides are widely distributed in nature. The I-glycone may be methanol, glycerol, a sterol, a phenol, or a base such as adenine. Also, glycosides are important in medicine because of their action on the heart. which is known as cardiac glycosides, and also because they all contain steroids as the I-glycone. Next, we're going to learn about amino sugars, um, specifically about the hexosamines. And here I have some information about it. And the first point that I would like to say is that amino sugars are components of glycoproteins, gangliosides, and glycosaminoglycans. They're also formed the replacement of one of the hydroxy groups of a sugar by an amino group or a substituent amino group. Um, Also, amino sugars include D-glucosamine, a constituent of hyaluronic acid, D-galactosamine, a constituent of chondroitin, and D-manosamine. Lastly, um, here we see that several antibiotics, for example, erythromycin, contain amino sugars, which are important for the antibiotic activity. Next, I'm going to be presenting you some information about maltose, sucrose, and lactose, which are important disaccharides. Um, the first thing that I would like to discuss is that the disaccharides are sugars composed of two monosaccharide residues linked by a glycoside bond. The physiological important disaccharides are maltose, sucrose, and lactose. And also, I would like to add that the hydrolysis of sucrose yields a mixture of glucose and fructose called invert sugar because fructose is strongly laboratory and changes the weaker dextrorotatory action of sucrose. And here I have an image about some disaccharide samples. You can see the structure and then the name of the example. And here I have some information about polysaccharides. And the first piece of information that I have is that polysaccharides are complex biocarbohydrates composed of long chains of monosaccharides, serving as crucial biomolecules of energy storage and structural support in various organisms. They're also essential in biochemistry, playing a key role in energy metabolisms, cell structure, and other biological processes. Also, here we see that starch is a homopolymer of glucose forming an alpha glucosidic chain called a glucosan or glucan. Lastly, we see that starch is the most important dietary carbohydrate in cereals, potatoes, legumes, and other vegetables. Next, I will be presenting you some information about glycoproteins, which are also known as mucoproteins. And here I have some important information about it. And the first one is that glycoproteins are proteins containing branched or unbranched oligosaccharide chains, including fucose. Also, they occur in cell membranes, and many proteins are glycosylated. Next, the sialic acid are N or O acyl derivatives of neuraminic acid. Neuraminic acid is a non-carbon sugar derived from monosamine. And lastly, I have the sialic acid are constituents of both glycoproteins and gangliosides. And the last big piece of information that I will be presenting you in this presentation is about the presence of carbohydrates in cell membranes and lipoproteins. And here I have that approximately 5% of the weight of the cell membranes is the carbohydrate part of glycoproteins and glycolipids. Their presence on the outer surface of the plasma membrane has been shown with the use of black lectins, which are the proteins that bind specific glycosyl residues. Next, I have that the glycophorin is a major integral membrane glycoprotein of human erythrocytes because it has 130 amino acid residues and it spans the lipid membrane with polypeptide regions outside both the external and the internal surfaces. Next, I have that carbohydrate chains are attached to the amino terminal portion outside of the external surface. And lastly, I have that carbohydrates are also present in A protein B of plasma lipoproteins. Here, I really wanted you guys to have like a complete image. It will be easier for you to kind of understand about the structure of the cell membrane if I give you a picture. And here you have this picture about here you can see like the inner and the outer surface of the cell membrane, and on the outer part we have the carbohydrate, glycoprotein, and glycolipid, and on the inner surface you can, for example, see the channel protein and the peripheral protein. This is just for you to have a clear picture of how a cell membrane will look. Okay guys, so here I have the summary with some of the most important information that I really want you guys to remember and learn. And the first piece of information that I have is that carbohydrates are major constituents of animal foods and animal tissues. They're characterized by the type and the number of monosaccharide residues in their molecules. Also, I have that glucose is the most important carbohydrate in mammalian biochemistry because nearly all carbohydrate in food is converted to glucose for metabolism. Sugar have large number of stereoisomers because they contain several asymmetric carbon atoms. And the physiological important monosaccharides include glucose, which is the blood sugar, and ribose, an important constituent of nucleotides and nucleic acids. Next, I have that the important disaccharides include maltose, an intermediate of the digestion of starch, sucrose, which is really important when it comes to dietary constituents containing fructose, and lactose in milk. And lastly, I have that starch and glycogen are storage polymers of glucose in plants and animals, and that starch is the metabolic fuel in the diet. Okay, so here comes the fun part. And here I have some multiple questions for you guys to kind of test your knowledge, the knowledge that I am presenting you in this, in all the slides that I have prepared for you. So the first question that I have says that which of the following statements best describe the focus of glycomics? So I have a few options for you, and I hope that you guys take the time to read the whole information that I'm presenting you and kind of be able to guess which one is going to be the answer to this question. The second question that I have is basically asking you which of the following carbohydrates is correctly matched with its role or characteristic. So again, I want you guys to pause the video and take the time to read through all of the choices that I'm giving you and come up with the best answer. Okay, so here I have some more questions for you. And number Number 3 says, which of the following statements accurately describe the presence of carbohydrates in cell membranes and lipoproteins? Number 4, which of the following pairs correctly matches a polysaccharide with its primary biological role? And again, I will like you guys to pause the video and take your time to read through the questions in order to be able to come up with the best answer. Okay, so here I had the last question, which is question number 5, and it says, which of the following statements correctly describes a type of isomerism found in glucose? And here at the end, I'm providing you all the answers in order for you to be able to compare with the answers that you come up with. So you can compare and see which one you got right and which one you got wrong. Okay, so I really hope that you guys got all the questions correct. That was the whole point of the presentation. Okay, so lastly, here I have all the references in case that you want to go back and learn a little bit more about this topic. I have listed all the references in numerical order that they appear in the presentation. So this is the end, and I really hope that you guys enjoyed this video and learn a little bit more about carbohydrates. Bye.